So I'd like to welcome everyone. Good afternoon. Welcome to the 9th webinar from SBBq. Today we'll have Dr. Aravind from the NCBI-NIH. It's a great pleasure to have him here. My name is Eduardo Reis. I'm first secretary from SBBq. I would just like to give you the welcome and quickly tell you that this activity is part of the annual meeting of the Brazilian Society of Biochemistry and Molecular Biology that's taking place in virtual format this year. And if you like to see the full program, you can follow the link that is shown now. And also inform you that this talk is going to be recorded and will be available to you for late presentation at YouTube, in the SBBq channel in YouTube. So without further delay, I'd like to pass the word to Tiago Venâncio from Universidade Estadual do Norte Fluminense, that will chair the session and will introduce you, Dr. Aravind. So,
thanks, Eduardo. Good afternoon, everyone. It's an enormous pleasure to me to have Dr. Aravind giving this web conference for all of us today. I will make a brief introduction of Dr. Aravind. In 2004, he got a bachelor's degree in microbiology at the University of Pune in India. After that, in 1999, he got a PhD in biology at Texas A&M University in the US. He worked for 3 years as a staff scientist at NCBI-NIH. After what, he became a PI at the same institute, and he has been working at NCBI for all these years. He has made enormous contributions to the evolutionary classification of proteins, including the discovery of dozens, if not hundreds, of conserved protein domains. He also has important contributions to the understanding of biological networks, and more recently on toxin and antitoxin systems. And he participated also in several important genome projects, such as the Human Genome Project. Over all these years, Dr. Aravind published more than 380 publications available in PubMed, and he accumulated over 95,000 citations that correspond to an H index of 136 and an H10 index of 330. So after this short introduction, I'm very eager to see the recent progress of his research. So Aravind, the word is yours. Thanks again for agreeing to participate of our conference.
Thank you very much, Tiago, for that very kind introduction. So let me go ahead and share my screen. Okay, do you see my screen?
Yes, you can go ahead.
Okay, so I'll be stopping my video and just having my slides on, just in case there's some bandwidth issues. Okay, so work in my lab has been focused on patterns. So now if I show you these two patterns here, they look very disparate, both look very pretty. If I tell you that by knowing this sequence of numbers and by performing some operation on them, you can generate both of these patterns, it might seem surprising to some of you all. And if you're curious, you can ask me at the end of the talk. This is not what I'm going to be talking about, but something related in biology. Here you have a nucleotide, here you have a protein performing some operation on DNA, and here is a sequence. Now, if I tell you that we have the means by reading the sequence to somehow get to the connection between these, it might again sound a bit surprising, but that's essentially what we have been able to get to in the past 3 decades or so of research. So how do we go about doing this, and why is this relevant? As you all know very well, today we have an overwhelming flow of data in the form of biological sequences and various other contextual information which comes on top of these sequences and structures. Now, the conventional methods of molecular biology and biochemistry, which were established more than 4 decades ago, which went gene by gene, are not exactly best suited to handle this wealth of information. While one may say that there have been several new systems or omics approaches which tackle this problem, still, as they say, the devil lies in the detail, and you cannot escape the detail when you're studying biology. So how do we make use of this great wealth of data to make biological discoveries? Now, in our science, we have only one bottom line, that's the evolutionary theory. So as long as you're able to apply the evolutionary theory, you can make sense in biology. So much of what we do is trying to apply the evolutionary theory to make sense. Now, in practical terms, what it involves is analysis of protein sequences and structures. And the caricature here is a very simple account of what we do. You may start with single sequences or whole alignments of homologous sequences, and we use this as an anchor to search protein sequence databases. And as a consequence of that, you can find more homologous sequences. You can condense the information which exists in them into something which we call a profile or a position-specific score matrix. And that is a statistical measure of, or a statistical representation of the conservation of a related group of sequences. And using that, you can search deeper to see if there are sequences which are closer to this profile as opposed to the random model, and detect deeper relationships. You need not stop there, and you can go ahead by comparing one profile against a library of profiles, and that offers an even deeper handle of finding more distant homologies. As a consequence, what we end up with is the discovery of the new domains, or the functional, evolutionary, and structural units of proteins. And this is joined by other forms of information, like today we have a great wealth of structure, so we can directly do this process through comparing structure similarities rather than by just comparing sequence similarities. And additionally, we can put on all the contextual information coming from the high throughput studies to add a further layer of functional picture to this relationship between protein domains. Now, one of the most powerful forms of contextual information, which applies primarily to prokaryotes, is that of gene neighborhoods or gene fusions. And in a sense, this serves as the famous Rosetta Stone, which is taken from archaeology, where for a long time the ancient Egyptian language was not readable with the extinction of the old Egyptian civilization. But the discovery of this inscription on stone called the Rosetta Stone, which provided a multilingual form of the same message, which had the old Egyptian language juxtaposed next to the Greek language, which people did know to read, allowed the decipherment of the ancient Egyptian language. So likewise, we have biological Rosetta Stones. Wet lab studies, careful individual biochemical or molecular biological studies may have established the function of, say, gene A. And what we observe is that in prokaryotic genomes, if two genes or two or more genes tend to be functionally associated, they tend to travel together in the genomes of organisms. That is, they are tightly linked into an operon. So by following the conservation of genome neighborhoods, we can establish a conserved gene neighborhood pattern. And thus we show A is functionally linked to B, which when we couple it with the sequence analysis, which I described previously, or structural analysis, we could arrive at a functional prediction for gene B. And the same thing applies to gene fusions, because this coupling typically goes with these genes forming complexes, their products forming complexes. Sorry. So the proteins which are, or RNAs which may be produced from these genes, they tend to form complexes, which reflects in these linkages. So this powerful form of functional inference, when coupled with sequence analysis, can give you a lot of insights into the functions of proteins which were previously unknown. So in a sense, this for us serves as a telescope. To use cosmological analogy, when you use more powerful telescopes, you look farther back in time. Likewise, the deeper you can look into the protein world, the better you can understand ancient relationships. And you can look deeper by finding these more distant relationships between protein domains. And one consequence of that is predicting new biochemistry for proteins with unknown functions. On the other hand, you have an offshoot of determining the flow of genetic information over evolution, both vertical as well as horizontal, and establishing the relationships in the tree, or what is more, the network of life. So around 3 decades ago, what started to crystallize from these studies is that cellular life form essentially has 2 major branches, the archaea and the bacteria. And it was within the archaeal lineage, and it was in course of the past 3 decades, we have understood this better, that a particular archaeal lineage known as the Asgard archaea gave rise to one of the components of the eukaryotes. Now, the eukaryotic mitochondrion, which is the other part of the eukaryotic cell, had a bacterial origin from the alpha proteobacteria. It was not just the cellular organisms which contributed to eukaryogenesis, but also the viruses which infected these organisms. So, in a sense, the eukaryotes can be seen as a composite lineage which combines both archaea and bacteria, as well as the selfish elements which had infected these ancient lineages. Now, that leads us to this more general concept of arms races in nature. Arms races can be seen across all levels of biological organization. The most visible ones are like prey and predator, and it goes all the way down to the microscopic level, where you have pathogens and cells in individual cells in these conflict interactions. And when you have such conflicts, what you see is an accelerated evolution of the molecules which are at the interface of this conflict. So it helps us study natural selection and evolutionary processes in the fast track. Now, these conflicts also merge at different levels. So, for example, you may have the predation of invertebrates by vertebrates, and it is likely that this mode led to the infection of vertebrates by what were originally invertebrate nidoviruses, which then radiated in vertebrates to give rise to viral forms, including the coronaviruses from which we are now kept at home. So this is the backdrop against which I'll tell you some stories of the discovery of new biochemistry. And the work which I'm presenting today was possible due to my present and former group members, including Robson, who is from the university, who is now at the University at Sao Paulo. And the work will be essentially presented in the form of 2 major stories which illustrate this interface between arms races in biology and evolution of enzymes. So before we get started, let's lay out the very basic material which sparked this study. So we have the central dogma, which is one area or one pathway of flow of biological information from the nucleic acids to the proteins, but not backwards. That's from proteins to nucleic acids. So this flow of information is primarily through nucleic acids. You have the genome itself, you have the transcription of the genome in the form of the mRNAs, and then you have various RNAs which assist in this process, which we can loosely call the translation RNAs, which include the tRNAs, and the rRNA, which catalyzes the very formation of proteins. Now, there's a very interesting symmetry between this core process of biology and what one may call a more peripheral process, which it's peripheral because it's not related to the replication of the information of life, but it is more related to physiology and homeostasis. And the symmetry between these two is that the information flow in these more peripheral or physiological processes for maintaining homeostasis also involves molecules related to nucleic acids, namely the nucleotides. So if we have nucleotides in this place and nucleic acids, one of the questions one may ask is, how is it that similar kinds of molecules came to be used in information flow in both these processes, although in somewhat different ways? Now, before we get to that, we should realize that one important thing associated with the central dogma. The central dogma implies the invariance of the code. And once you have something invariant, it becomes a target for attack in biological conflicts. So we have codons which are more or less invariable across whole of life. There are some variations, but for a good part, you have the same type of coding, whether it's E. coli or a human. Now, when you have the same type of coding, then that becomes an excellent target for something which can attack it in a biological conflict. And what we see is that indeed, the ribosomal RNA, the tRNA, and the like are excellent targets for enzymes deployed in biological conflicts. And evidence for this comes from the fact that across all levels of life, be it vertebrate toxins or plant toxins like ricin and abrin, or microbial toxins such as colicins, which are used in conflicts between two members of the same species, like two different strains of E. coli, or fungal toxins. The common denominator in all this is the attack on nucleic acids and the flow of information through the central dogma. And the most prevalent mode is the hydrolytic attack, where there is the breakage of the phosphodiester bonds in the nucleic acids. The alternative mechanism is through toxins like ricin, which break the glycosidic linkage to clip off the base, but the end result is similar, which is crippling of the flow of information through the central dogma. So this is not restricted to purely offensive processes. It's also a part of defense or the immune system, and other mechanisms such as addiction, like toxin-antitoxin systems, selfish elements which cause themselves to be addicted or cause a cell to be addicted to them, or if they are born on a plasmid, they cause the cell to be addicted to a plasmid. Now, the way they elicit this addiction is through a toxin, and the typical toxins are endonucleases, which target tRNAs or rRNAs in certain cases. And a very similar set of toxin like RNases are also seen in a wide range of antiviral systems, both in eukaryotes and prokaryotes. One example is RNase L, which is one of the most potent antiviral RNases in vertebrates. So coming back to this thing, we have the flow of information through the central system, through nucleic acids and the flow of information through the periphery. Now let's look a little bit closer at this flow of information through the periphery, and I'll lead from here into a very interesting connection to biological conflicts. So the information flow through nucleotide-based signaling can be very simply represented through this cartoon, where you have some kind of a stimulus. For example, in our visual system, we have the stimulus of light, and eventually this leads to the production of one of many types of nucleotides, or alternatively, it could be a derivative from a nucleotide. Like in plants, you have this signal in the form of a cytokinin, which is the nucleobase, a modified nucleobase clipped off from the nucleotide. And that serves as a signal which is eventually sensed, and that sensing leads to a response. Now, in biochemical terms, we can break it down into three broad classes of protein domains. You have the synthetases, which synthesize the signaling nucleotide, and sometimes they are called cyclases, like the adenylyl cyclase, because the nucleotide is a cyclic nucleotide. And then you have something which has to sense the nucleotide, which may be nucleotide sensing domains most commonly. And in some cases, you have specialized ribozyme like RNAs, which sense this, more precisely riboswitches, which sense the nucleotide. And on the other end, you need the termination of the signal. For the shape of a signal is very important if we want to have a signal in the first place. And that's mediated by phosphodiesterases, which break down the phosphoester linkages in these nucleotides. So one of the early activities in my group was trying to discover more of these nucleotide sensing and generating enzymes, as well as phosphodiesterases. We discovered a bunch of these through sequence analysis. And eventually a common theme came to fore, which was very striking. It became clear that nucleotide synthetases have emerged at least twice in evolution from the same common ancestor as nucleic acid polymerases or nucleotidyl transferases. So like, say, the primary polymerases which synthesize DNA as part of replication in our cells, any eukaryotic cell, have this conserved domain which is known as the RNA recognition motif, RRM-like fold, which is also called the palm, because the early structural biologists saw the polymerases as holding the nucleic acid like a palm of the hand holding something. And so this palm domain, which is found in these nucleic acid polymerases, replicative nucleic acid polymerases, like the eukaryotic primary replicative nucleic acid polymerases, they are related to many of the nucleotide synthesizing enzymes and signaling, like the adenylyl cyclases, the guanylyl cyclases, and the GGDF domain, which generates the cyclic diguanylate. So this is one fold. Now there's a distinct fold which is not related to this beyond the fact that they have a 4-stranded core sheet. And this one is the Pol beta fold after the mitochondrial DNA polymerase beta. And it's also the fold found in the primary replicative DNA polymerase of bacteria. That's DNA polymerase 3. And this fold is shared by another class of nucleotide synthesizing enzymes and signaling, like the 2', 5' oligoadenylate, which is involved in interferon signaling. So this interesting connection sparked our interest to see what may this relationship between nucleic acid synthesizing enzymes and nucleotide synthesizing enzymes. And and this is where the connection to the biological conflicts came, because one thing was clear, that some of these nucleotide synthesizing enzymes produce key signals which activate immune response in vertebrates, like cGAMP and 2,5-A. And when we are looking at it in this view, this is where the active site which binds the two magnesium ions and synthesizes the nucleotide is located. But when we turn it around by 180 degrees, you find that there is a region on the rear side which has these conserved patches of residues, and they are typically basic in nature. And we realize that this is the site which is actually recognizing the nucleic acid and stimulating the catalytic activity of these enzymes. So one thing which was a big mystery until about 6-7 years back was, where did these vertebrate signaling enzymes come from? So what we found was that in bacteria, there's a large group of related enzymes, which we call the SMODs, for the second messenger oligonucleotide or dinucleotide synthetases. And when we analyzed them, we found that in addition to the active site, they had the same type of conserved pattern in the rear end, suggesting that they are also stimulated by the binding of a ligand, which is likely a nucleic acid. And when we performed the evolutionary analysis, we found something quite interesting, that they are lodged deep within this radiation of enzymes, which are involved primarily in central dogma related processes like RNA repair and the generation of the 3' CCA end, which is required for the aminoacylation of tRNAs, or the poly-A polymerases, which add the poly-A tail to mRNAs. So we can infer from this that the common ancestor was performing these RNA processes, and it was within the radiation of those enzymes, these signaling enzymes which are involved in immunity in animals at that point emerged. So one question which came to fore was, what were the bacterial SMODs doing? If the eukaryotic ones are involved in immunity, that's the animal ones, can we infer something about the bacterial ones? So for that, we decided to do an evolutionary analysis going backwards systematically. First, we looked at all the versions in eukaryotes, and we found many new versions outside of vertebrates, which are typically in the form of what are called lineage-specific expansions, or several paralogous copies per genome. For example, in this oyster, you have 80 copies, and they come in many different families, which could all be unified. And remarkably, we found that these expansions are associated with some variable regions found in the C-terminal part of it. So this pattern is very suggestive of a role in biological conflict, where rapid evolutionary variation in a paralog family typically is associated with some kind of immune pressure. So we got a hint that even outside vertebrates, these are involved in some immune processes. And then when we proceeded to bacteria, we were able to use the method which I mentioned earlier of contextual analysis. And what we found were the genes for these SMODs were invariably coupled to a second gene. And a closer examination showed that many of these second genes had a very characteristic organization in terms of the protein they produced. So they had a constant part, which was a domain which we were able to identify called the Savade domain. And they had a highly variable region, which was typically to the N-terminus. And when we looked at this variable N-terminus, we found that there was these were globular domains. They were not just low complexity regions. Each was a distinct globular domain, and we were able to unify many of them to enzymatic domains which were previously found in other systems involved in immunity, like the restriction endonuclease fold domain, or the HNH domain, which is a new RNase or DNase domain, which is again found in restriction enzymes as well as other systems which target nucleic acids as part of immune mechanisms. Further, we found bacterial homologs of the caspases, which are associated with apoptosis in eukaryotes. In particular, apoptotic processes can also play a role in immunity, for they limit the spread of an infective agent. And another example of this intersection was the TIR domain, which was for the toll interleukin domain, interleukin receptor domain, which was again previously recognized in eukaryotic immunity. And now we could connect it to these bacterial proteins which were associated with these SMODs, nucleotide transferases. So what do you make of this pattern? We were able to condense all of this into this flow, wherein you have a stimulus which is likely in the form of an invasive nucleic acid, which leads to the SMODs nucleotide transferase or nucleotide synthetase, generating a signaling nucleotide, which might be a cyclic nucleotide as in animal case. And that is in turn sensed by the sensor domain, which may be the Savate domain. And that activates these associated enzymatic domains, which are essentially attack domains, which may attack the invasive entity through nucleic acid degradation or peptidase activity, or they initiate an apoptotic process or program cell death to limit the infection. So thus, we had evidence that these bacterial homologs of what were the eukaryote of what are characterized first in vertebrates as signaling agents in immunity also likely play an immunity role. So this role in the immune response could be pushed back to the ancient bacterial world. Now, there is more to this, but I'll just briefly mention that not all systems are just two genes. Some of them have some further wrinkles to them. Just one example which I'll talk about is that some have this more rudimentary version of a ubiquitin conjugation system with just an E2 and an E1 ubiquitin conjugating enzyme and a JAB deubiquitinase. And this kind of a system is likely to play an auxiliary role in addition to the primary effectors which are associated with the SMODs nucleotide transferase. Now, moving on, we found yet another interesting connection between nucleic acid polymerases and potential signaling nucleotide cyclases. Now, this came about from this mysterious polymerase called TUG1, which had been characterized in tRNA maturation and repair. Histidinyl tRNA, for example, has a nucleotide at the -1 position on the 5' end, which is added in some cases in a template dependent and in other cases in a template independent manner. So how is that added? It's added by this enzyme TUG1. It can not only add one nucleotide, if you give it a template, it could add more nucleotides. And it happens to be the only only polymerase, nucleic acid polymerase, which goes in the 3', 5' direction, as opposed to all the typical polymerases which go in the 5', 3' direction. So it was a mystery as to how this whole thing functions. And through sequence analysis, I was able to show that this is actually related to a class of minimal DNA polymerases, which we found in certain viruses and plasmids. And thus, it suggested that there was a unified mechanism of both the 3', 5' and the regular 5', 3' nucleic acid polymerases. And an examination suggested that they have a similar set of catalytic residues, and we proposed a mechanism by which they may catalyze, TUG1 may catalyze this unusual elongation at the through the addition of a nucleotide through the 3' 5' mechanism. So what did this lead to? The understanding that TUG1 has a palm-like nucleic acid polymerase fold allowed us to look further into this family. And we were able to unify TUG1 with a very enigmatic enzyme, which we had first reported many years ago in what was one of the earliest accounts of what's today famous as the CRISPR systems. So a subset of these CRISPR systems contain a large enigmatic protein, the CRISPR polymerase protein, which looked like polymerases, but it was not clear what they were doing. Now, the unification of it with the TUG1 polymerase suggested that it was a likely nucleotidyl transferase. And we found very minimal versions of it in what could be termed the ancestral or precursor minimal versions of these CRISPR systems with the polymerase. And here you had just the polymerase domain coupled with a nucleotide sensor domain, which we had earlier described, called the CARF domain, and a nuclease domain, or a pore-forming toxin domain with 2 TMs. Alternatively, it may be, this is a gene fusion here, and I'm showing the protein coming from the gene fusion, or you may have a conserved gene neighborhood where you have a linkage similar to how the SMODs and the SAVADE protein was, and the CARF is in place of the SAVADE. Now it turns out that CARF and SAVADE are related nucleotide sensing domains. So this allowed us to get a handle on these mysterious non-type 2 CRISPR systems. So this is an earlier classification of the CRISPR systems which we had described, and these are particularly abundant, this polymerase domain, in this type 3 systems. And the architecture is with a HD domain, which is a phosphodiesterase domain, an inactive polymerase domain, and then an active copy. And then they are linked to these with CARF and CARF containing effectors. So when we look at it across CRISPR systems, it became clear that this is a very distinctive feature of the type 3 systems. And we were able to thus bring them under the same umbrella as the SMODs-based signaling. So the idea here is similar to the SMODs-based signaling, where you have the stimulus in the form of the invasive nucleic acid. And you have these CRISPR polymerases which generate a nucleotide signal. And that in turn is sensed by the CARF domain, which triggers the associated effectors, which may either attack the invasive entity directly or trigger cell death to limit the invasive entity. And finally, you have the signal termination, where some versions of the CARF domain are catalytic and they hydrolyze that nucleotide signal themselves, or it is controlled by the HD phosphodiesterase, which hydrolyzes the signal. So what we see here is that many disparate nucleotidyl transferases have been incorporated into an immune signaling mechanism. So this realization allowed us to come up with a synthetic model to explain the potential relationship with the central dogma. So this is the idea which we have, that right from the primordial nucleoprotein world, a variety of central dogma processes, like say amino acylation or nucleic acid repair, or both RNA repair and DNA repair. And these repair events happen due to direct attacks on nucleic acids in biological conflicts. They generate as a byproduct certain peculiar nucleotides or RNAs with cyclic nucleotide ends, which comes from metal-independent cleavages of RNA. And these byproducts served as the initial signals that the central dogma information system is under attack. And that in turn allowed a response to take place. Like it gave us a sense that there was a biological conflict underway. And hence there was selection in two directions. One where you had these becoming repair enzymes in a dedicated way. And in the other direction where they gave rise to signaling enzymes, which signal the attack. And some of these became eventually dedicated signaling enzymes, which may be involved in general signaling processes outside of biological conflicts. So this is the first story. Now I'll move on to in the last part of my talk, to the second story. And this relates to a completely different kind of issue, again, connected to immunity, but a very different kind of nucleic acid modification. Now, what we see is that as the life of an organism increases, and if an organism has circulation and a mucosa, it's very likely you encounter the same pathogen over and over again. So there is going to be natural selection for some kind of adaptive immunity or an immune memory, which can be stored in a protein-based system, unlike CRISPR systems in prokaryotes, which store it in the nucleic acid. So the way it's done in vertebrates has been very well known for a long time now. But just to summarize it, is you mutate self in order to discriminate non-self. So what's this mutation of self? This mutation of self is carried out by an enzyme called the deaminase, which deaminates cytosine in situ in DNA, resulting in uracil. Now, uracil is not tolerated in DNA. And so there is a repair process, which is error-prone, and it induces mutations in DNA. This mutagenic process is required for the recombination, triggering the recombination events, which result in immunoglobulin diversity generation, as well as hypermutation, which specifically cause antibodies to increase in their affinity towards an invasive entity by selection for more avid binders. So both these mutagenic processes are acting on self DNA, but they help to increase the specificity for non-self, and that's detect non-self. So one big evolutionary question was, from where did this enzyme AID come into being? Because it's a dangerous enzyme for a cell to possess when you're mutating self. So earlier studies had shown that this process of antibody diversification, it exists all the way till the base of the jawed vertebrates, where you have the 8 deaminase as well as the recombination process, the VDJ recombination to generate antibody and TCR diversity. But there was a mystery of what happens in the more basal vertebrates, the jawless vertebrates. And these jawless vertebrates were once a great radiation, but today they are only represented by two lineages, the lamprey and the hagfish. The lamprey being a parasitic fish which feeds off other fishes. And studies by Zeev Panzer and Max Cooper had shown that these jawless fishes, they also possess lymphocytes, just like vertebrates, and like the jawed vertebrates. And they found that these lymphocytes express a certain gene called the VLR gene, which in the embryonic state is in a disassembled state. So it suggested to them that there was a parallel process going on, but these VLR genes are unrelated to the antibody genes or the TCR genes with immunoglobulin domains in the jawed vertebrates. So the question was, is there an AID ApoBec-like enzyme which mutates the DNA for diversification jawed vertebrates in these jawless vertebrates? So that's when Panzer and Cooper collaborated with us, and we were able to find two AID ApoBec-like enzymes for the first time through sequence analysis of the lamprey genome. And we propose that these might be the versions which are involved in this process. And subsequent experiments by Panzer's lab showed that indeed that was the case. He was able to express those in vitro and show that they had a characteristic pattern of cytosine mutation. And further, we found that by analyzing these VLR genes, which are not antibody-like genes, but they're comprised of these repeat elements called LRRs, that they have a diverse, they have a region where diversification happens in these three rims across one surface. And subsequent solution of a crystal structure with an antigen by Roy Mariuza's group, with whom we were collaborating, showed that it was indeed this surface which bound the antigen. So that helped us push back the APOBEC-like enzymes, mutagenic enzymes, to the common ancestor of all vertebrates. And because the antigen receptors are different, here a VLR in the case of the jawless vertebrates and an antibody in the case of the jawed vertebrates, it suggested that these antigen receptors emerged after the mutagenic system was already in place. But there's still the question of how far back these go. And that question, the answer to that question came from a very unexpected quarter, where, which came from a study of nucleases. We have been long interested in discovering new nucleases through sequence analysis. And this study took a deadly turn at some point when we found that a whole class of nucleases are present in are present in bacteria, which are not typical of, say, DNA repair or holiday junctions or RNA processing, because they had signals which were suggestive of them being secreted, and they were associated with domains typically found in secreted proteins involved in adhesion, like the laminin G or the ricin domain, and various lectins and fibronectin type 3 and so on. And what was more interesting was that we found they had a characteristic organization where this nuclease domain always came at the C terminus. And these proteins were highly variable, with the N terminus being constant, but the C terminal nuclease domain may widely vary. And further analysis showed that it was not just nucleases, but the C terminal tip may be replaced by deaminase domains in some cases, peptidase domains in others, and as a result, we discovered a large number of such alternative domains. Now, this led to the important prediction that these are likely secreted because of this tripartite organization, where the conserved N-terminal has domains very characteristic of particular secretion systems. They're absolutely diagnostic for these systems and not found anywhere else. And then they have a central region which may include region modules or a linker part which forms a superstructure. And finally, you have the C terminus where you have these nuclease domains. So what you see is an extensive shuffling of a constant N-terminus or relatively conserved N-terminal part with a variable C-terminal part. The other interesting thing we found through genome context analysis was that many of these are linked to a second protein in their vicinity. And one such example is the Suk domain, which through careful analysis we found has a pocket which shows the maximum diversity, whereas the rest of the protein is quite conserved. So this led us to the idea that this accompanying protein is probably an immunity protein for the nucleases and other effectors encoded by the other gene. Furthermore, these genes had been earlier characterized as recombination hotspots, but nobody knew why they are recombination hotspots. And looking at the data with this new information which we had, we were able to show that these recombination events correspond to various cassettes which comprise of simply the toxin and immunity modules, recombining with the core gene, bringing back a new toxin or an immunity protein pair. So that explained that C-terminal diversity. So this led to a simple model wherein we predicted that these are likely toxins which are secreted or presented on the cell surface, which are protected with the protein, with produced by the gene accompanying these protects the cells which produce them. And if there is a match, that is, if the two produce the same, then they can go along. But if there's a mismatch, the tips are different and the immunity proteins are different, there'll be a killing interaction and there'll be exclusion. So this is a good example whereby you can exclude non-kin. And exclusion of non-kin in bacterial biofilms and colonies is of particular importance because you don't want cheaters using the shared goods such as siderophores and secreted enzymes which are produced. Now, as I said, in addition to nucleases, we found several deaminases. And this is an example of the wide deaminase diversity which we found in these toxin tips. And that allowed us to finally find 8 apobec members outside of the animals, outside the vertebrates. And we found that in this vast radiation of toxin deaminases, there were the first non-animal, non-vertebrate versions of these deaminases. And a closer analysis allowed us to recover them in eukaryotes outside of vertebrates, like in the most basal animals, as well as some non-animal lineages like these algae like Micromonas and Emiliania. And the smoking gun of that they were likely transferred from bacterial systems came from these endosymbionts like Wolbachia, where we found in their secreted toxins an 8 apobec like deaminase tip. So, and also in other intracellular pathogens like Orientia, which is the agent of typhus. So putting all this together, we were able to show that there was a much greater radiation of these 8 apobec deaminases, well beyond what had been recognized. And we performed a systematic analysis identifying all of those, including secreted versions in eukaryotes. And to summarize, we were able to synthesize this into what we term the lymphocyte hypothesis. So the lymphocytes are a cell type which replicates very actively. It has a high proliferative capacity. Hence, it's a good target for viruses, especially retroviruses and HepaDNA viruses, which can infect these. And they kill 2 birds in one stone, as they say, because they both limit the immunity by infecting the lymphocyte. And on the other side, they get a cell with a lot of proliferative capacity, which is a good one for a virus. So what we propose is that these bacterial deaminases through lateral transfer were acquired by animals and expressed in these lymphocytes because they served as a mechanism to counter these retroviruses by mutating their DNA. Thus, what was once a part of the biological conflict in bacteria now became an armament in the animal lymphocytes. In the next step, we proposed that some of these started evolving more slowly. They got institutionalized, and instead of mutating the retroviral DNA, they started mutating self-DNA. And as a consequence, we now had these mutagenic enzymes which could be deployed against self molecules and allow them to diversify, thereby leading to adaptive immunity. So this is what we call the immunocyte hypothesis. which culminates ultimately in these adaptive immune systems in eukaryotes. So here we have yet another example of where bacterial immunity has led to, or bacterial biological conflicts has led to an adaptation which was used in vertebrate immunity. And on a parallel note, we saw earlier that these systems, such as the SMODS-based nucleotidyl transferases, which have parallels to the type 3 CRISPR systems, are essentially mechanisms which first emerged in bacterial immunity, and they were used as is in eukaryotic immunity. So if an arms race is taking place elsewhere in the tree of life, the armaments which are generated in that can be quite easily ported elsewhere and used in immune mechanisms in some other system. So that's sort of the lesson which we learned from this analysis. In the interest of time, I guess I'm quite past time. Let me stop here and take any questions you may have.
Thank you very much, Aravind, for this very thought-provoking talk on deep subjects on the protein evolution. I will open for questions now. People may use the raise hand function of Zoom, or can post the question in the question and answers box.
So I'm not seeing the questions if there are any. Could you? Are you seeing them?
I will read the question for you. There is one question in the question and answers box from Teca Galvão. She says, hi, with respect to the organization of genomes and operands, what would you say is the current paradigm? For example, for two-component systems, they are often, but far from always, in the same operand. Would you suggest that this is a way to keep doors open to functionally relevant cross-signaling?
That's a possibility. So there actually the operon organization is very finely affected by various selective pressures which may result from things like cross-signaling, or in cases where you need the molecules, the proteins, to diffuse away from each other. You don't require them to be in a strong complex, in a tightly bound complex. In that case, they may break apart. But there is another form of contextual information which usually does allow you to link things even in this situation, which is the phyletic patterns. So if you have parallel phyletic patterns, then you can see that those two are likely going together in the genomes. And because we have a very large number of genomes today, those phyletic pattern vectors are also very effective contextual information.
Mm-hmm. Okay. We also have a hand raised here from Emmanuel Passarelli. I will open his microphone.
Hello, everyone. Dr. Aravind, thank you so much for your very inspiring talk. My question is regarding the toxin and antitoxin system. Mm-hmm. One of my recent works, we were exploring population structure of a bacteria, Klebsiella aerogenes, and mm-hmm. one of our main results was one plasmid that was characterized by these toxins and antitoxin systems. So mm-hmm. The main result was that this plasmid was in a very frequency in the population that we studied. More than 60% of the isolates harbor this plasmid. So mm-hmm. when we tried to explore this toxin and antitoxin system, we only found the toxin. This plasmid was not well characterized. So my question for you, what would be the best approach to find this, or to explore more this this information that we have. I think that we were handling with RNA antitoxin system, but I'm not sure. I'd like
So
your
you say
opinion.
there was only a toxin and there was no antitoxin?
No, I said that we could easily find the toxin, but the antitoxin was not clear. So
well, yeah, so the RNA is an option because we were amidst the first to report an RNA antitoxin in collaboration with Stewart's group at the NIH. So it was a situation which might have been similar to yours because what she had was a toxin, but the antitoxin was not clear and the toxin activity could be detected when expressed by itself. But under natural circumstances, there was something keeping it in check. And it turned out that it was a RNA encoded. The RNA was being transcribed in the opposite direction from the toxin itself. So an RNA-based regulation is a possibility. Now, it is also possible that there are certain antitoxins which are low complexity and poorly conserved, like the ones which come with these ADP ribosyl transferase toxins. And sometimes those genes are not translated. And if you're not done a translation yourself of the neighboring region, then that's a possibility. A third thing is that it's elsewhere on the plasmid. Since your system is plasmid borne, if the plasmid is sort of addicting itself, the two need not be coupled as long as they are there together on the plasmid, but at a slightly more distant position.
yes, but the plasmid was very small. It's like 90,000 database base pairs, and I knew that was a colicin type of toxin.
Okay.
Co-RNA1 type, I think.
So,
but
okay, so in that case, it's even possible that there is no antitoxin, and actually a subset of cells are sacrificing themselves to produce the toxin. Since we do see that with colicin, where there is a lysis of a subset of the cells. Not all cells express it, a subset sacrifice themselves in order to make the toxin, which provides a collective advantage to the whole group.
Okay. Thanks, Emmanuel. We have two more questions here. One from the Chuck Farah. Regarding the nuclease activity of the AHH domains that you mentioned near the end of your talk, do you have any information regarding the specificity of their targets, DNA, RNA, sequence specificity? I ask because we have identified this domain associated with P4SS effectors involved in bacterial competition.
Yeah, so a priori, we cannot be sure if it is purely DNA or RNA. There have been some people who we had some discussions with who claimed it was a DNA, but we cannot be sure because their results look very bad, to put it simply. Now, this AHH toxin, it belongs to this larger group of enzymes known as the HNH-like superfamily. Members of the HNH-like superfamily, the majority of them have been characterized as DNases, but a few of them have been characterized as RNases. So we cannot rule out RNase activity entirely, though I would personally lean towards DNase due to the preponderance of DNase activity amidst HNH enzymes.
Okay. We have another person asking to talk, Guilherme Gomes. Go ahead, Guilherme.
Hello, Professor Aravind. First of all, it's an honor to even talk to you and ask you a question.
Oh, so, that's too much.
I'm a student. I'm a student in Hobson's lab, so
okay.
I have read lots of your work, and I'm very fond of your work.
Thank you.
I've seen the distribution in Crassostria of a domain, SMODS domain, I believe. And I'm finishing up my master's degree right now, and I worked with the GTPAs, and I was working with the GMAP domains. And I have seen the same distribution, not the same, but an LSC-like distribution of the GMAP domains also in Crassostria. And I wanted to ask, first of all, how can we differentiate between these LSCs from the whole genome duplication that happens in these animals? Because I have also seen this distribution in fishes. And another question is, why is this happening with these organisms? Why is this happening in Crassostria specifically? Because I cannot see these distributions in other animals too, other than fishes.
Okay, so the question has two parts to it. One is the whole genome part, which is more easily answered. So if whole genome duplications don't usually lead to large LSEs. So if you have an LSE which is like 3 or 4 members, that could arise from ploidy. But we don't see any evidence for, say, ploidy of 80 times. So if you have a very large number, it's almost always an LSE which has happened specifically of that gene. And the second way to test that is by looking at genomic locations. You won't find any other kind of synteny with associated genes. These will be scattered all over the genome. That's a very typical signal of a LSE which is happening independently of ploidy. Now, the second part of your question is why in this organism? We have systematically surveyed LSEs in the past, and we have an old paper on that which you could look up. Each organism has its own LSEs. It's just that different organisms have different lineage-specific expansions. For example, in plants, you may have one type of lineage-specific expansion of transcription factors, like the WRKY transcription factors. But you won't see any of that in animals. So by definition, depending on the selective pressures which an organism has faced, you see very different sets of lineage-specific expansions. Now, what we do know, and we have a paper in the works in this very regard, that in certain marine organisms like Crassostria, they do tend to show expansions in immune-related molecules. You see that in the sea urchin too. So this is like an alternative to having antibodies. So if you have in the genome many copies, you can, in a sense, produce that diversity, which is produced through recombination in, say, TCRs or antibodies, by just expressing different alternative versions which may interact with different invasive entities. Does that answer your question?
Yeah, thank you very much, Professor.
Okay. Now there is a question from someone that you might know, Aravind, Dr. Robson de Souza.
Okay.
He's around and wants to ask a question.
Well, good to hear from him.
Hi, Alan. How are you?
Not bad. How are you, Robson?
I'm fine. Great to see your presentation. That was a delight.
Well, you were part of some of these, so you know this well.
Yeah, but it's great to see it all put together. Great. Well, I was going to ask you more about something more towards the end of the presentation, because I was curious that the ApoBecs that you mentioned, the deaminases, well, they have they don't seem to be, there are both some with the genetic activity, they may cause mutations anywhere, and you have the ApoBec proper, they seem to target the immunoglobulins, the exons of the immunoglobulins domain.
Right, that's 8. That's 8, and the ApoBecs are more generally mutagenic.
Right. And then when you have systems like that are diversity generating systems, you have a certain, in many cases, you have for the immune systems, you have this directionality. But and I have seen about bacterial type, which is the DGRs, they are called DGR, diversity generating heteroelements.
Yes.
Also more specific. They tend to target certain genes, as far as I understand.
That's correct. Because they have a template and they do it through a reverse transcriptase mechanism.
Right. So you think, well, you know about other mechanisms, diversity generating mechanisms that work like that? Of course, there was a long discussion about the role of the SOS system towards generating more diversity, but it was more not directed to like those systems.
Right, right. Okay, so DGRs, I don't know if you saw our paper which came out earlier this year in eLife. So if you have not seen that, take a look at it. In that we describe reverse transcriptase based DGR systems which we believe are involved in prokaryotic immunity, which have a very interesting tripartite organization. So they have two constant components, which are essentially an AA plus ATPase, and which the chaperone, MOX are like AA plus ATPase, and its co-chaperone, which is a VWA domain. And there is a third component, so these two are constant, and there's one more component, that's why we call them the ternary systems, called the VMAP, which is VWA MOXR associated protein. Now, this VWA protein, it has at its C terminus one or more domains which show remarkable diversity in the bacteria which possess them. Now, in cyanobacteria, these are coupled to a reverse transcriptase, and they are clearly undergoing this DGR-like diversification. So here is one example of yet another more directed diversity generating system. Then we have these systems in mollusks, which are called the FREP proteins, for fibrinogen-related proteins. And they seem to undergo somatic diversification, which is parallel to what the TCRs and immunoglobulins undergo. So these are some additional examples of DGRs, so to say, if that answers what you wanted.
The FREPs are directed at our specific targets too.
Yeah, so it's only the FREPs which undergo mutagenesis, apparently not others. The FREPs are expressed in the lymphocyte equivalents of these snails. And they did a sequencing and they found that the FREPs undergo diversification, but others are mostly, other genes don't. So we identified an aid apobec-like deaminase in the mollusks, which we suspect is involved in the diversification of these FREPs. That's reported in the deaminase paper. Now, in terms of aid, it looks as though aid has some chromatin cofactors, which specifically limited to those regions which are involved in, which are the exons of the immunoglobulin genes or the class switching regions which are upstream. So the limiting to those regions seems to be by these other factors, which are probably well conserved ones because they simply retain open chromatin domains in lymphocytes.
Just to close the questions, if we had time, I was going to ask very briefly if you would expect more systems like these to exist yet to be discovered.
Oh, absolutely. We have a paper in the works.
Oh. Okay.
Which actually I should have submitted, but you know, it'll take me maybe a month more to finish, hopefully.
Thank you, Robson.
Thanks, Robson.
Thank you.
We are running out of time. I have the two last questions in the questions and answer box. I will make them both and you can reply together if you prefer.
Okay.
The first question is from Wesley Barrios. Thank you very much for your presentation. Do you think that DNA and RNA aptamers can be an ancient form of defense prior to proteases? And the second question is from João Paulo de Almeida. Do you think that non-coding RNAs that have evolved together with toxins such as antisense RNAs in type 1 antitoxin systems would also have a recognizable pattern of evolution?
Okay, so for the first question, that's an interesting thought, and probably such mechanisms did exist in a very early phase of evolution. And one could imagine that there is some survival of that, but the evidence that they are still active is not that clear, at least to me. But that they functioned, that they were at the interface of conflicts in the very early universe, early biological universe, that's definitely a possibility. Now, the second question, could you repeat it so that
if non-coding RNAs that have evolved together with toxin systems.
So the problem with detecting fast evolving non-coding RNAs is that unlike proteins, you are working with just 4 bases. And when you have just 4 bases, and finding homologies in RNAs can be very difficult. So I do suspect there are a lot more antisense or antitoxin RNAs lurking out there. And we simply are not detecting them because we don't have the handle of homology. But we get a hint that they do exist because when you look at some of these solo toxin genes, you see what we call a gray zone next to it, where there is no coding, protein coding entity, but you have a large enough gap in an otherwise fairly dense region, dense in terms of protein coding, which suggests that perhaps you have an RNA being produced in that gap, from that gap.
Mm-hmm. Well, I would like to thank again Dr. Aravind for the superb talk. I would like also to thank the attendees for coming, pretty remarkable attendance. Before we go, I would like to pass the word again to Eduardo Reis for some announcements.
Thank you, everyone. Just like to join Thiago in thanking Dr. Aravind for his time and giving this excellent, outstanding presentation for us.
Thank you.
And I would just like to announce, to show you the next activity of the virtual SBBq meeting. That is, hopefully you can see it now. That's gonna take place next week. Will be about education of biochemistry and molecular biology in times of COVID-19. So hope you can join us in this meeting too. So with this, I think we close the session, and thank you, Thiago, for sharing this event, and everybody stay safe and have a good time.
Thanks, Aravind.
Thanks, stay safe, bye.
Bye-bye.